I'm Camilla Scheele, Associate Professor here at CBMR, and it's my great pleasure to introduce our first speaker, Dr. Zoltan Arany. He's a Samuel Bellet Professor of Cardiology and the Director of the Cardiovascular Metabolism Program at the Perelman School of Medicine at University of Pennsylvania. Dr. Arany has an educational background from Harvard, and he has advanced through many prestigious fellowships and research positions, and he's now focusing on mechanisms that underlie metabolic pathophysiology in the cardiovascular system. So let's give him a warm welcome. Please take the floor, Joel.
Thank you. Okay, here we go. So thank you for inviting me here. It's a real pleasure to be here. It's my first time in Copenhagen. This is very exciting. I want to spend the next half hour or so telling you a little bit about some of the work we've been doing in the space of human cardiac metabolism. It's not the only thing we do. This is my lab slide. I've been accused of being unfocused, which I think is true. We're a little bit all over the place, but I'm going to try to focus today on just talking about human cardiac metabolism, some of the work we've been doing in the last few years. Let's see, does this work? No. Oh yeah, it does. So the context that I want to put this in is heart failure, and I realize that I think I'm the most basic talk today, so hopefully I won't bore you too much, but we're going to do a little bit of molecular biology here. So the context that we think about is heart failure, because it's still a problem in the United States and everywhere else in the world. Whether it's reduced ejection fraction or preserved ejection fraction, at least in the US, if you're admitted into the hospital with one of those diagnoses, your 5-year mortality is still about 50%. It's actually improved a little bit lately, but that's not good. That's not all that different from pretty severe cancers. So we've got lots of treatments, but they're not sufficient. And I would argue that except for maybe a couple of newcomers, most drugs that we use to treat heart failure are not actually directed at the cardiomyocyte itself. They're actually directed at the vasculature and neurohormonal status. So there's opportunities in thinking about what's going on inside the heart, and that's what we like to think about. So we like to think about metabolism, of course. That's why we're here. The heart is logical to think about metabolism. It's the most metabolically active organ in the body. Its per unit mass oxygen consumption is tremendously high. It's more than any other tissue, and it's pretty dynamic. So you can actually increase it quite a bit with exercise or other stressors. So much so that I don't know how many of you have ever held a heart in your hand, but if you do, about a third of what you're holding is actually mitochondria. We think of it as a structural thing that pumps, but it's actually 1/3 of it is actually just generating energy for the other 2/3 to work. So it's incredibly metabolically active, and there's lots of things that can go wrong. And there's pretty decent evidence, and I'm just showing you one example, but there's actually hundreds of papers that support this notion that there's a problem with energetics in a failing heart. My favorite kind of evidence is this kind. This is just a review article from Stephen Neubauer from a few years ago, but this is non-invasive NMR-based evaluation of energetic status in patients who have heart failure versus patients who don't. And you can tell by the ATP to phosphocreatine ratio that there's an energy charge deficiency in failing hearts. And in fact, it's an extremely good predictive biomarker, if you will, has predictive power to determine whether you're going to do well or not in the next few years. So there's a problem with energy in the heart. Why is there a problem with energy in the heart is one question that many people have wondered about. So I'm going to sort of try to get through these three things. If we don't have time, we'll skip the last bit. The first question that we were very interested in is what fuel does the heart use anyway? And so we'll talk about that, and then we'll take a peek inside the heart, and then I'll talk about branched-chain amino acids if we have time. So what fuel does the heart use was the first question that we became very interested in. And actually, we're not the first to ask this question, of course. There are literally hundreds of papers out there that have asked this question. And if you can Google it, and you'll get a review article. I'm picking on my friend Gary Lopashuk, but I could have picked on anybody else. Usually the reviews look something like this. And the take-home message is that the heart is called an omnivore. It can really use lots of different fuels, fats primarily, but also glucose and lactate and amino acids and ketones. And then some of these things, I don't mean for you to look at the details of this slide too much. And then some of these things go haywire in heart failure. Typically, people talk about the fat to glucose shift, in heart failure. I'm sure many of you have heard about that. There's an increase in use of ketone oxidation. There's an increase in use of glucose, but a decrease in glucose oxidation. So there's this notion that the heart's a little bit Warburgy in heart failure. But we looked at this a few years ago, and I realized, our lab realized that, and when I say the majority of study, I mean 99+% of studies to generate this review are done in rodents, right? Sometimes pigs, sometimes dogs, but it's really all rodents. So how do we know that any of this is true in humans? So let me introduce, for the non-cardiologists in the audience, the concept of a catheter. I'm sure it's not a shocking concept. So this gentleman, Theodor Otto Worsman, in 1956, won the Nobel Prize for the idea of putting a catheter through a vein and thread it into the heart to be able to get blood from the heart or visualize the heart. Actually, it was so controversial at the time that no one would let him do it. And he eventually did it to himself in order to prove that it was doable. There's a very funny anecdote around that. But this is the original first picture of a catheter. It's him. There were no such thing as cardiac catheters at the time, so this is a urinary catheter that he threaded up his arm. Now, we're a lot more sophisticated these days, of course, and we can put the catheters in all sorts of sophisticated places. And the place of interest for us was the coronary sinus. Right? Why the coronary sinus? Because the coronary sinus drains the heart and only the heart, which basically means that if we can simultaneously sample blood from the artery and from the coronary sinus, we can see what the heart is doing. Right? And if there's more of something in the coronary sinus, that means the heart spit it out. And if there's less of it in the coronary sinus, that means the heart used it. Very simple notion. We're not the first to invent it. There's about 50 studies done in the 60s, 70s, and 80s, as soon as that catheter was invented, that have looked at this. But there's not hundreds of those studies, and they sort of stopped happening in the 2000s. And we can talk about why that is. I have a few ideas. But most of the studies are pretty rudimentary. They're using old technology. So we wanted to revisit this with modern technology, meaning mass spectrometry, right? So this was a fantastic collaboration that we did with electrophysiologists in our group, David and Matt. Chalsoon, who used to be a student in my lab, now has his own lab, did all the most of the mass spectrometry. And it was all led by an amazing MD PhD student, Danielle Morshiga. And so the notion is patients are coming in for an elective procedure. They're on the table. We draw blood from the coronary sinus because the electrophysiologists are there anyway, and we simultaneously draw blood from the artery. It's really simple, actually, conceptually. This was published a few years ago. I just want to give you a few of the highlights and contextualize it. So, the data looks like this. I don't mean you to squint. I'm just showing this slide. Just so I'm sure you've all seen volcano plots before. The y-axis is the p-value, log p-value. The only reason I'm showing this slide is to show you that the data set is really good. Right, we're getting multiple hypothesis corrected p-values in the 10 to the negative 10. So not for every metabolite, of course, but for quite a few. So the data is really good, but don't squint at this. Let me just summarize it for you. I'm going to skip all the math and all the analyses and just give you the punchline for the sake of time. So we can calculate all the metabolites and the fuels that are taken up by the heart. We can quantify them. And then we can do the conversions through standard chemical reactions as to how much each fuel is contributing to ATP generation in the heart with a few assumptions that I'm happy to talk about. And so you end up with a pie chart that kind of looks like this. So this is the relative contribution of each fuel to ATP. In yellow is free fatty acids. And then you've got quite a few amino acids, quite a few ketones, and quite a bit of lactate. The heart is a net lactate consumer. People tend to forget that. It might spit out a little lactate, but it's actually a net lactate consumer. And then you have this gray thing, which is what we couldn't account for, which we are almost certain is actually fat that is coming from lipoproteins, because we couldn't measure those for technical reasons. So you can see, and again, these are an important point. These are fasted individuals, right? They're coming for an elective procedure, so they haven't been eating. So it turns out one of the things that I'm not showing you here is the heart is extremely catabolic when you've been fasting overnight. Much more catabolic than the leg. We did the leg at the same time, and the heart is much more catabolic. And I don't understand why that is, or what purpose that serves. If people have ideas, I would welcome them. But this is sort of rough picture of the average patient that was on our table, and the fuels that they're using to generate ATP. So again, the large majority is fat. There isn't an accounted oxygen consumption, which we think is lipoproteins most likely. Net consumer of lactate, avid consumer of ketone for sure, and there's active proteolysis. Note the absence of glucose. I always get a question about that. We'll come back to that in a minute. Now, of those 120 or so patients that we did, 40 of them had reduced ejection fraction, right? And so was there a difference between the ones who had normal function and the ones who had reduced ejection fraction? The answer is yes, there was. First, you'll notice that glucose didn't appear. So we're not seeing this famous fatty acid to glucose shift, at least not in this patient population under these conditions. But we are seeing less fat use, definitely and significantly less fat use, and it's being picked up by ketones and lactate for the most part, some amino acids as well. So there's some sort of shift going on, at least in these subjects. So going back to the review that is based 99% on rodents, we're not quite seeing the same thing. We're not seeing this massive increase in glucose use. We're not seeing a decrease in glucose oxidation. We're not seeing a decrease in lactate oxidation. In fact, we're seeing an increase in lactate oxidation. So some things are the same and some things are not the same. And it's worth thinking as to why the things are not the same. It's not just because it's mice versus humans. It's also because the humans are sick, or many of them are not sick, but are just the average 60-year-old American. The average 60-year-old American, represented by our group, has a BMI of 30, right? Has 10% of them smoke, 15% of them are diabetics, 15% of them are hypertensive. So that's not the average mouse that we're studying either. So the point I'm trying to make here is that when we go back to look at our mouse studies, we try to make sure that we are modeling what we see in humans before we ask our mechanistic questions. That's the point I'm trying to make there. But so this was quite interesting. But the obvious question then became, why, or how rather? How is it that in heart failure these changes are occurring in humans? And we can't tell that from an arterial to coronary sinus study, of course. So then we turn to the only way to answer that question is to look inside the heart instead of before and after the heart. And so we moved to trying to do that. That's a little more complicated because most patients are not excited about sharing pieces of their heart. So what we did is we teamed up with Ken Margulies, who's a heart failure doc at our institution and works with the transplant folk. And for the last literally 25 years has been collecting post-transplant hearts. So patient goes to have their heart transplant, heart is removed, usually goes to the garbage can, but not in our institution. In our institution, it goes to Ken. He also gets, and this is very important, normal hearts. How does he get normal hearts? Because donors arrive, so somebody has a motorcycle accident or some other unfortunate event, and the heart arrives for the transplant surgeon to take, but then the surgeon doesn't take it for some reason. It's too small or something like that. So then we get it. That's a discarded donor. So we have normals, which makes all the difference in the world. Importantly, these hearts are harvested extremely carefully and they undergo cardioplegia before they're ever taken out of the body. This is super important. The heart is so metabolically active. I don't know how many of you have worked with heart in the mice in metabolism, for example, but if you take the heart out of the mouse and you leave it on your bench for 30 seconds, everything changes. Right? You can't do that if you want to study metabolism. So here, everything underwent cardioplegia before it ever got ischemic. So very important. Yeah, we always take it from the same spot. And then we compared basically, so obviously I want to make the point, this is end stage, obviously. This is end stage heart failure. So that's a caveat to this study. And then we used non-ischemic dilated cardiomyopathy for the sake of homogeneity. We've actually gone back and done ischemic cardiomyopathy and have the same results now. But I'll just show you what we recently published with non-ischemic. So again, you get these beautiful volcano plots with excellent p-values. So these are robust data sets because modern mass spectrometry is so great. So there's a lot of data in this study, but I'm going to contextualize it in the context of the first study. Okay, so in the first study, I showed you. Oh, sorry. First sanity check. Is there an energetic defect in failing hearts? And the answer is yes. There we find exactly what you could see non-invasively by NMR. We see reductions in ATP. We see reductions in ATP to phosphocreatine ratio, so we see exactly what we predicted in terms of energetic status. For the aficionados in the audience, it's interesting that we don't see any change in NAD or NADH, which has been kind of a big debate in the field. We're not seeing that. So, okay, I'm going to talk about 4 fuels. I could talk about endlessly about all the different fuels, but I would bore you to death if I'm not doing it already. I'm just going to talk about 4 fuels. And again, I'm going to contextualize it in the context of the first study. So I showed you in the first study that there's a decrease in failing folk, who have failing hearts. There's a decrease in fat use. So why is it that there's a decrease in fat use? Can we figure it out by looking inside the heart? So just a quick reminder of how fat oxidation works. I'm sure you all know, but fat enters the cell through transporters. It gets ligated to a large, bulky hydrophilic adduct, coenzyme A. That's stuck in the cytoplasm. You have to get it into the mitochondria. The way you get it into the mitochondria is through the carnitine shuttle. So the CoA gets removed, carnitine gets put on. You go inside the mitochondria, and then you swap back the carnitine for a CoA again, and then you undergo fatty acid oxidation. Clip off 2 carbons at a time. The party line in those 99% rodent studies, and the data is really good in rodents, is that there's a problem with oxidation. So there's a problem with this process in failing hearts in rodents. So if that was true, you would expect that there would be a pileup of all the stuff upstream. You'd have more acylcarnitines, you'd have more acyl-CoAs, you'd have more fats. In fact, what we see is the opposite. We see massive reductions of acylcarnitines in the tissue, massive reductions of fatty acids and fatty acid CoAs. Everything is down. Everything is down upstream of fatty acid oxidation. So I think the problem is up here, somewhere up here, somewhere in fatty acid transport, import into the heart. And I don't have the answer. I don't know how this is happening, but I think it's pointing us to a different spot than we've been thinking about in all of our rodent studies. And we're thinking more about that. So that's fats. What about lactate? So now we have more lactate coming in. Why is there more lactate coming in a failing heart versus a non-failing heart? So when we looked at these hearts, we didn't just look at metabolites. We also we did proteomics, we did RNA-seq, as you might imagine. We just threw our omics book at it. So the answer for lactate, I think I know, based on what we saw. So again, a reminder of glycolysis. Glucose comes in and it goes down glycolysis, you end up at pyruvate, or lactate comes in directly, ends up as pyruvate, enters the mitochondria, and the rate-limiting step is the pyruvate dehydrogenase complex, which is regulated by an inhibitory kinase. I think most people remember this. And what we see in the hearts is a dramatic reduction in the kinase. It's the #1 most reduced protein in our proteomic set. So if you have a dramatic reduction in the kinase, then you would have dephosphorylation of the complex, which is what we see. You would have activation of the complex, because remember, this is an inhibitory phosphorylation site, which is what we see. So what I think is happening is the spigot is open. That's why we're having more lactate. Right? Now, what's regulating the kinase, I can't tell you. But at least I think I can explain why there's more lactate going into a failing heart. The spigot is open. This is actually important because people have argued that this is a good thing to have more lactate going into the heart. And there's actually even been a couple of small trials that have tried to give a kinase inhibitor to the heart to improve cardiac function in humans. And my argument, based on these data, would be that that's pointless because the spigot's already open. You can't open it anymore. So it's an important observation. Ketones are also increased quite a bit, tripled. Why are ketones increased? Here, I think the answer is completely different. It has nothing to do with spigots. I think ketones basically just go in direct proportion to how much ketone there is. So I don't think the heart regulates its ketone use. It's just proportional to how much it sees. Right? There's no regulation. And the reason we know that is because the tissue levels are elevated, the plasma levels are elevated, and the amount of ketone that we see is directly proportional to the levels in the plasma from patient to patient. So we don't think there's any regulation here, it's just that there is more. But this also is important because this suggests that the more ketone you give, the more ketone the heart's going to get. And there's good data that giving ketones is beneficial. So the prediction here would be that it's going to work. We'll see down the line if it does, of course. And then lastly, I always get this question, why is there no glucose on this picture? Right? And I think it's an interesting question because there kind of ought to be. And I think there's multiple potential answers. The first is, as I told you, they're fasted individuals. So the insulin levels are low. We're actually repeating this study. We can't feed them because they're coming in for elective procedures and the anesthesiologist won't let us give them food. But we can infuse nutrients. So we're repeating this study now, infusing nutrients. So next year or whatever, next 2 or 3 years, we'll have the answer to that question. But I actually think the answer is going to be no, because I think the real answer is these patients are hyperlipidemic. And many of you may have heard of the Randall idea of Randall cycle, which is the idea proposed more than 50 years ago by Randall, that at least in the heart, there's inhibition of carbohydrate uptake when you have a large amount of fatty acids uptake. The mechanisms for it are complicated and still somewhat controversial. But there's good evidence for it. Even in people, this was a really elegant study done 35 years ago now, where they took, again, exactly the same thing as we did, arterial and coronary sinus sampling to see how much glucose uptake is occurring in individuals. And then they gave them fat. They infused fat. And as the fat level increased in the arterial, in the artery, the glucose uptake by the heart went down. And it sort of got to zero around when the fats were around 800 micromolar, right? Now, where are we, right? Where is the average American in his 60s on our electrophysiology table? Almost all of them were above 1 millimolar, right? We have a lot of fat floating around in our bodies, at least in the United States. I can't speak for Denmark, but we're pretty bad. So I think this is going to be the answer, but we'll see. We'll see when we do the studies with infusions. So again, I just focused on 4 fuels, but taking these 2 studies together, you can spend hours and hours looking at these 2 studies and learning about what's going on with cardiac metabolism, normal and heart failure. It's been quite a lot of fun. There are caveats, as I said. These are fasted individuals. We're following it up with infused with nutrients. They are from end-stage hearts, so I can't really say what's going on in a heart that's making its way to end stage. We're just extrapolating from these data. And all these data are in heart failure with reduced ejection fraction. So we're trying to think of ways to kind of address these caveats. One of them is collaborating with other folks. So we did a couple of really nice studies working with either David Cass and Virginia Hahn at Hopkins, where we looked at biopsies from HFpEF patients. So they actually biopsy their heart failure with preserved ejection fraction patients, which is very uncommon. And it's interesting because their patients are extremely obese. Their average BMI is 40 in this study. And despite the fact that they're extremely obese and they're extremely hyperlipidemic, we saw exactly the same thing as we did in the other hearts, which is massive reductions in all the acylcarnitines and acyl-CoAs in the heart. So even in that context, there appears to be a block in fatty acid uptake, right? And the same was true in a study that we did in hypertrophic cardiomyopathy subjects, where the septum was surgically excised, because sometimes that gets in the way, and we see exactly the same thing. So I actually think that this problem with fatty acid import into the heart may be an early event, and it may be general to many forms of heart failure. So again, this is why we're very much interested in thinking more about this. How am I doing for time? Okay, 10 minutes, good. So that's the kind of human studies that we've been doing, and as I mentioned, we're doing many more. But of course, we're interested in causality, and you can't prove causality with in human studies for the most part. So we're interested in taking these observations, and it's not like we don't like rodents. We do like rodents, and we do like genetic studies in rodents. So we of course are going back and doing causality experiments in rodents based on the observations that we see in humans. And I'll just show you a couple of vignettes. We'll see what I get through. But I do want to reiterate the point that when we do these studies, we as much as we can, we want to make sure that we are modeling what we're seeing in the humans when we do the study in the mice. Otherwise, I would argue the mouse study is a lot less interesting. So one of these pathways that we've been studying for quite some time is the pathway of branched amino acids, which I purposely didn't show you the data for the humans. I'll show it to you in a second. So just a quick reminder, branched amino acids, I'm sure everybody knows, leucine, valine, isoleucine, they're essential amino acids. You can't make them, you have to eat them. If you eat too many amino acids, too many leucine, valine, isoleucine, you can't store them either, you have to combust them, right? So if you have a big steak, as we are wont to do in the United States for dinner, you don't wake up in the morning with 12 more ounces of muscle. It doesn't work that way. You burn it. It would be nice if it worked that way, but it doesn't. And it all gets burned in the mitochondrial. And I won't walk you through the pathway, except to say that the rate-limiting step is this complex, PCKDH, which is equivalent to PDH, pyruvate dehydrogenase complex. And much like PDH, it's regulated by a kinase, an inhibitory kinase, BCKDK. So just remember that, because it'll matter in a moment. So here's the data from the hearts, and many other people have noticed this as well, right? So if you look at a failing heart, what you see is a reduction in most amino acids, except for the branched amino acids. Those are elevated. We're not the first to see this. Other people have noticed this before. And in fact, if you do RNA-seq and proteomics, et cetera, you see reductions in all the enzymes in that pathway that breaks down branched amino acids. And so this has led to the hypothesis, the dogma, I would even say, in the field, that there's a catabolic defect for branched amino acids in failing hearts, and that's contributing to heart failure, right? Now, is this really true? Well, we don't think so, because, well, first I'll say that consistent with this hypothesis, if you reactivate branched amino acid catabolism, you benefit heart failure models, right? So if you have a small inhibitor, which many people use, BT2, of that kinase, so if you're inhibiting the kinase and dephosphorylating the complex, you get more flux through that pathway, right? You're inhibiting the inhibitor, more flux through the pathway. And if you do that, the plasma levels of branched amino acids go down. That makes sense because you're combusting them, right? And now you use models of heart failure, and you can use many of them. Here's one of them: myocardial infarction, surgical infarction of a mouse. You improve outcomes. Their ejection fraction is better over time when you give them this drug. And you can use, many labs have done this, at least half a dozen labs all across the world using various different models. This is highly reproducible. So it fits the model. The problem is, the assumption here is that it's working through, the drug is working through the catabolic pathway in the cardiomyocyte, right? It's activating the cardiomyocyte catabolic pathway. So we wanted to test that. And the way to test that, you can't give the drug just to the cardiomyocytes, of course, but you can delete the kinase in just the cardiomyocytes, and that's the same thing, basically, right? So we made new flox alleles because they didn't exist to the kinase, and we deleted it just in the heart, and you can see that, you know, exactly what you predicted in the heart. The phosphorylation is gone, the flux is active. We measured the flux is activated, and now we give a myocardial infarction, and nothing happens. Nothing at all happens. We did this every which way we could. It didn't work. So then the question became, okay, if it's not in the cardiomyocytes, where is the benefit? Because the benefit is undoubtedly happening when you systemically activate the catabolism, but it's not working through the cardiomyocytes. So we looked at skeletal muscle, we looked at liver. To make a long story short, I think the answer is the vessel. Back to the vessel, just like we started in some ways. It turns out that when you activate systemic branched amino acid catabolism in a mouse, you lower blood pressure and you increase vascular reactivity. And we've done this many different ways, by telemetry, by tail cuff, lots of things. It's interesting that the pathway is independent of nitric oxide. I'm sure you all remember Nobel Prize nitric oxide released from the endothelium vasodilates the smooth muscle cells. That is not how this is working, because you can neutralize the nitric oxide pathway and you still get the vasodilation. And it's vessel autonomous, so it doesn't even have to be in the animal. You can give the animal these drugs, and the vessels are less constrictive. You can even just take a wild-type vessel and give the drug, and the vessel will dilate. So there's a direct effect of catabolism on these vessels. And we think, now this is all work in progress, but we think it's actually through the smooth muscle cell itself. And recent work by Rio, who joined the lab not too long ago, has shown that indeed, if you delete the kinase just in smooth muscle, you lower blood pressure. And if you do the opposite, you delete the catabolism. DBT is one of the key enzymes in the catabolic pathway. So you block catabolism instead of inducing it, you increase blood pressure. We don't yet understand how this works, but this is basically telling us that, you know, based on that the dogma is wrong. Basically telling us that these benefits in heart failure models are probably not working through a direct effect on cardiomyocytes, but instead working through vasodilation. And the last thing I'll say about this vignette is that we start with human. We make observations in mice that were unexpected. Are those observations at all relevant to humans? We try to go back to humans as much as we can. And we've done a couple of studies. I'm just showing you one. This is a Mendelian randomization study, which I think in some ways is as good as it gets for causality in humans without actually doing a drug study. So Michael Levin, who's now junior faculty at Penn, basically used polymorphisms in GWAS studies that basically predict who is going to have higher levels of branched amino acids. and then used those polymorphisms to interrogate the UK Biobank to see if they related in any way to blood pressure, right? So that's basically a Mendelian randomization. And the answer was yes. All of them, especially all the SNPs that were close to enzymes that made sense, such as the phosphatase and the kinase, impacted blood pressure. And a couple of other people have recently also gotten similar results. So there's clearly a relationship between branched amino acids and vascular reactivity and blood pressure in humans. And so we're trying to understand this. This was recently published. Some of this was recently published, and some of it is not yet published. So if you want more details, you can look there. So that's one example, and that's one more detailed vignette. I'll just end by telling you a couple of other things that we're doing in the lab that relates to these. So we've become extremely interested in understanding systemic metabolism and cardiac metabolism, and now we're deep into doing steady-state heavy isotope infusion studies, because this is a great way to really understand quantitatively metabolism in a rodent. So you infuse you're at a steady state, your carbon-13 fuel of choice, glucose, leucine, whatever you want. You allow it to achieve the steady state, and then you sacrifice the animal. And so from the steady state, you can calculate turnover, you can do math, and then when you sacrifice the animal, you can ask, where do the carbons go? So if we're doing a glucose infusion, which tissue is using that glucose? It's a very useful and powerful technique. And we're using it in the context of heart failure. I'm just going to throw a pitch out for because I know this crowd's interested in thermogenesis. We recently just published, Mark just published a story where he basically did this in mice at room temperature versus cold and used every single fuel you can imagine, one at a time. And so this is a comprehensive picture of metabolism flux in response to cold in a mouse. There's lots of really interesting observations in there. If you're interested, you should go check it out. And we also have a study that's actually, so Jesse figured out how to do this in running mice, which impressed me to no end. So this is a mouse that's got two catheters sticking out, right? It has a catheter in its carotid and it has a catheter in its jugular. We use the carotid to draw blood. We use the jugular to give blood. And all of that is happening as the mouse is running, right? It's pretty powerful. So that's under review. So you'll see those data very, very soon. And I don't have time to show them now, but there's a lot of really interesting observations in there. So back to heart failure. So one of the things that Jay noticed in these data sets that I was showing you is that in the heart, in the human hearts, there seems to be a problem with single carbon metabolism. I won't go through the whole pathway of single carbon metabolism, but in essence, methionine is used as a single carbon donor, and it's incorporated into the acetyldenyl methionine cycle and used for things like histone methylation and various other methylations. And there's clearly a problem in the human failing heart. All these levels are down in the human failing heart. And so what Jay is now doing is going back into the mouse, using the infusion approach to quantify quantify how methionine is used and how much methionine is used in the heart and elsewhere in the body, for that matter, and making new floxed alleles to the different enzymes in this pathway to try to understand where a block can cause heart failure. So this is the paradigm that we are. And the last thing I'll say is the other cool thing that we're doing now is we're also using Ken's hearts, the human hearts, and doing these infusion studies. So this is a piece of a human heart. Nick has been leading this. This is a piece of a human heart that's getting perfused with heavy isotope label fuels that we can then trace into the TCA cycle or whatever. So I'll end there. So it's sort of the paradigm of how we're approaching this, right? We don't think that a human is the same thing as a mouse. We have a lot to learn from the mouse, but we do think it's different. And so we try to do as much as we can in the humans, and I showed you some of those examples. And then we go back to the mouse for mechanistic studies. That's the paradigm that we're trying to follow. And if you're going to remember two things from this talk, I would ask you that you remember these two things. First, we think there's an early defect in fat intake into the heart in models in actual human heart failure. Right? I would welcome input on what you think might be causing that, because we don't know. We have ideas, but we don't know. And then this cautionary tale about branched chain amino acids. which might be extrapolatable to many other things. We have a dogmatic view of what's happening based on observations, but when you test them, we got something completely different, right? You know, the idea, the branched amino acid observation was that you have reduction in catabolic enzymes, and therefore you must have reduction in catabolism. I didn't show you the data, but that's actually not true. It turns out that in heart failure, there's more branched amino acid catabolism because metabolism isn't dictated by just enzyme levels, right? It's dictated by substrate availability, by product inhibition, by post-translational modifications. There's all sorts of things that we cannot measure in cell culture that we cannot measure by doing enzymatic assays, which is why we think doing in vivo isotopic infusions are so powerful, because they tell you what's actually happening in an intact organism. So I'll stop there and welcome questions. I have the most awesome team. We have a total blast, and this is the best part of doing science is the team. Thank you.
Thank you so much. That was a really beautiful talk.
Thanks.
So yeah, I'm fascinated about this defect in fatty acid import. So how about the actual adipose tissue that surrounds the heart and the epicardial fat? Do you think there could be something with the signaling there?
It's possible.
What are your thoughts about
it's
about
possible.
that?
We were talking a little bit about this yesterday. So there's no, I don't think there's any clinical evidence that I know of changes in amount of epicardial or pericardial fat in subjects with heart failure. There is some interesting evidence that the intramyocardial triglyceride pool is altered in heart failure, but that's not the question.
No, I was also thinking about the actual adipokines that can be secreted from the adipose tissues. That can have an effect on the fatty acid import.
Yeah, it's possible. I don't know of any data that speaks to it, but it's definitely possible. We also don't see any correlation. We tried to do all sorts of correlation in this study to say, okay, does the amount of fat uptake correlate with levels of free fatty acid, or does it correlate with BMI, with adiposity of the subject? We didn't find any correlations asking those questions.
Okay.
But that doesn't, that's just conjecture. It doesn't really. It's possible.
Very interesting.
It's possible.
I think we have several questions from the audience.
Yeah.
Do we have a microphone? Yeah.
Hello, Umur Keresh from Lund University. So thank you for the amazing talk. So my question is regarding this lipoprotein-derived ATP. So in the body, as far as I remember, there are many tissues that have lipoprotein receptors, and they can uptake the lipoprotein as a whole molecule rather than hydrolyzing them into free fatty acids.
Yeah.
So do we know how heart is using that kind of fuel to use to generate ATP?
Yeah, it's a great question. I don't think we do. If I had to bet, I would say it's really the large majority is lipoprotein lipase. Now, as you know, the lipoprotein lipase sits on the luminal side of the endothelium. Even though it's made by the cardiomyocyte, it's secreted and transported across the endothelial layer and lives on the luminal side. The heart is the largest, has the highest expression of LPL of any tissue in the body. So I suspect it's mostly LPL mediated. And if you delete LPL, you can block fatty acid, lipoprotein fat uptake into the heart. There's some really elegant work done by Ira Goldberg many years ago that addresses that. So I think at least in mice, it's mostly through LPL and not so much through entire lipoprotein intake. But having said that, that process is active in the heart, uptake of entire lipoprotein particles. So that's an active area of study. But I think quantitatively, it's going to be mostly LPL mediated.
Is the free fatty acids in that region of the heart is the same with the rest of the body, or could the free fatty acids that you measure in the heart could be the hydrolyzed free fatty acids from the lipoprotein, or?
You mean in our arterial coronary sinus
exactly,
study?
Yes.
Well, in the arterial coronary sinus ACS study, we're measuring free fats in the artery and free fats in the coronary sinus. Right. So Now, maybe what you're asking is, are any of these fats, especially in the coronary sinus, a consequence of spillover, if you will, of LPL activity in the microvasculature? We can't tell the answer to that. So I suspect that some of it is the case. The only way to know that is to use labeled fat. So if you infuse labeled fat in a human and see the difference in enrichment, if you will, in the artery versus the coronary sinus, that would tell you whether some of it is getting spit out. And actually, that's been done. Back in the 80s, there's a couple of studies that did that and showed that there is active spillover under some conditions. So it's a great question. I think in the end, it doesn't matter to our math. I'm happy to talk more about that. But I think there's some of that going on. It's a great question.
Hi, thank you for a nice talk. My name is Erik Richter. I'm from the University of Copenhagen. I was just wondering, I was thinking about the analogy to skeletal muscle. Because if a skeletal muscle is stressed with high intensity exercise, lipid oxidation goes down, we know that. And one of the reasons might be that carnitine is sequestered by acetylcarnitine, so there's less carnitine for the CPT1. So you can't import the fatty acids. Do you think that's a similar mechanism that is going on? Have you measured carnitine?
Yeah. We have measured carnitine and it's not down. So that's the first answer. But I also don't think that that's the. I also don't think that's the mechanism, because if that were the mechanism, again, we would see a pileup of the upstream stuff. We would see anyway more cytosolic acyl CoAs or even free fats in the cytosol. If the problem was with the carnitine shuttle, and that's not what we see. We see reductions in everything, including free fat in the inside the cardiomyocyte. This is just based, it's a static observation. I can't prove about the flux, of course, but it's less consistent with that idea. That's a good question.
Hi, great talk. Dipshika Biswas from CBMR. I would like to know your thoughts about the branched-chain keto acids. Do you see differences in your human study? And also, one could argue that under certain contexts, like in BCAT knockout mouse, where the branched-chain amino acids are quite high and the BCAAs are low, we do see protective effects in terms of insulin signaling and glucose metabolism. So what are your thoughts on that?
Complicated question. In the human hearts, we see elevations in the keto acids as well as we do in the amino acids. So that's the sort of static answer to your question. Now you're bringing in the fascinating fact that there's a very clear relationship between branched amino acids and insulin sensitivity at the organismal level. And that seems to be true in humans, and it's definitely true in mice. So I don't know if this is exactly what you're asking, but is there a relationship between insulin sensitivity and heart failure? That may be how the branched amino acids are affecting the heart failure. Is that, you're nodding your head? So I'm going to say that's your question. We don't know for sure, but it doesn't seem to be the case, at least in our mouse models. Because we can modulate, if we modulate we've actually knocked out these enzymes in every tissue we can think of. Liver, muscle, as you saw, smooth muscle, heart, and the phenotypes don't travel together. The heart failure and the insulin sensitivity don't travel together at all. It's still early in the game, but I don't think that's how it's working. Oh, are the keto acids more important than the amino acids in the phenotype? Well, given that we still don't completely understand the mechanism, I don't think we know. So for example, the vasodilation that we see when we knock out the kinase, that could be because we're reducing the keto acids. We don't know that yet. Those are ongoing experiments. We don't know that yet. And your point about some of this can be answered by modulating the transaminase, which is what you were suggesting, and those are the kinds of experiments we're doing now. It's a great point.
Hi, really interesting talk. Just 2 short questions. So first off, we see these elevated lactate levels that you've observed. and then you pinpointed that it was down to this kinase. Do we know of any clinical implications or any known mutations, for example, from heart failure patients that are more prone to developing this condition that may be linked to this, and we might be able to see a clinical effect?
Yeah, that's a great question. I don't know of any polymorphisms or any mutations in the kinase that relate in any way to heart failure. But remember, this kinase is ubiquitous.
Right.
Right, it's used by almost every single tissue to regulate its glucose uptake. The effects of any variant in the kinase are going to be pleiotropic throughout the human.
And just one last remark. So this branched amino acid story and what you've shown, you've of course tested this in advanced heart disease models. And of course, by the time we get to heart failure, a lot of things will happen, right? The metabolism will change. There will be accumulating conditions where you end up with this chronic disease effect. Are there any known models, or are you working on this, of having maybe an early stage model that we can, or can we even study this, a transition, patients that are maybe more prone to developing this condition, that we can help to really understand what is actually going on, right? To get to that stage.
Yeah, that's it's a really important question. So I reiterate the caveat that all the tissue data I showed you, almost all of it, is end stage. Right? And you could argue that's a burnt out stage and it doesn't tell you anything about what's going on early. The two studies that I showed you at the very end, the biopsies from HFpEF patients, heart failure with preserved ejection fraction patients with the Hopkins group, and the myomectomies from the HCM patients, hypertrophic cardiomyopathy patients. Now, those are not end stage, right? Those are compensated patients who are coming in not in actual clinical heart failure and lying down on the table and having their procedure. So I would argue those are earlier events, and we see very similar things in those as we did in the later ones. But it's only two little snippets. I think a lot more needs to be done. The last thing I'll say is that in a lot of mouse models, we don't see this. So the whole PDK4 thing, PDH regulation, could be very different in humans versus mice.
We have one last question. Okay, we have two last questions, if it's brief.
Following up on the question, I was wondering what is the implication of the fuel consumption in maybe earlier stages of heart failure to preventing the deterioration of heart failure?
Yeah, I mean, that's a fantastic question. That's kind of the overarching hypothesis of the entire research program, is that we can find things that we can modulate. We haven't proven it yet. I think the most promising thing right now, I would say, not necessarily based on our work, is ketones. It does look like giving ketones to the heart helps the heart function, and there's been even a couple of human studies that have supported that, including a study where subjects with heart failure with reduced ejection fraction were infused ketones, and there was demonstrated improvement of cardiac function. It's a hard study to interpret because the ketones also affect the vessels, right? The interaction between the heart and the vascular system is strong, and so it's hard to tell what's chicken and what's egg in the effects you see. But I think at this stage, if I had to put money on something, I'd put it on ketones. But that's just why we do this, because we think there may be other things. I know that's kind of a hand-wavy answer.
Would the comorbidities like type 2 diabetes or obesity affect fuel consumption as well?
So that's also a fantastic question. We tried to ask that a little bit in these groups of patients. Some of them are diabetic, but the numbers are small. So the short answer is we don't know. We tried to see if there were differences between the diabetics and the non-diabetics, and we didn't see anything statistically significant, but the numbers are too small. We're continuing to do these studies, so hopefully as we have more numbers, we'll have an answer for you.
Okay, it has to be a short question. Final question.
Hi, thanks. Simon from Steno Diabetes Center. I enjoyed your talk very much. I'm also working on metabolism and metabolomics. I have a question. Can you a little bit elaborate more on the technique you use? Does it may contribute that you don't see the glucose, since you may use a C18 column where the glucose is not retained?
Now you're asking more than the average PI can answer. We use a variety of different columns. This is done with more than one column, and we definitely pick up glucose. Although I agree with you, glucose is not always easy to pick up, and it's much noisier than other metabolites, interestingly, on LCMS. But we're using LTMS, we're using C18, we're using other columns as well, both positive and negative. So all of these were done 4 different ways.
Okay,
Thanks.
let's thank
Thank you.
Zoltan once
Thank
more.
you.
Fantastic talk. Thank you so much. Get this gift.
Thanks.
Thank you.